How does an ELISA kit actually work? What's the difference between sandwich competitive and direct formats? What are recovery, linearity, and CV? How do you pick the right kit? And what if you want to build your own kits? Come along, Gil walk you through all of it in the simplest way possibly. We'll break this down into bite-sized episodes. First up, how an ELISA turns a color change into a concentration. Picture a single drop of blood that contains tens of thousands of different proteins. Picking out just one and counting every single molecule would be mission impossible. Is there an easier yet still reliable way to know how much of that protein is in the sample? Absolutely, our goal is simple. Find the concentration of one specific protein. Everything else is background noise. The sample itself is the unknown. Let's flip the problems. Pose. We already have the same protein at a known concentration. If we let the unknown and the known react under identical conditions, they give identical signals. Then, by definition, their concentrations must be equal. Congratulations! You've just grasped the core logic of ELISA. That's why ELISA is called relative quantification. In practice, we run a set of standards with known concentrations, measure their optical density OD, and plot a calibration curve, OD versus concentration. Slot the OD of an unknown into that curve and its concentration pops out automatically. That's why ELISA is called relative quantification. So what exactly is an OD value? Stay tuned. Next episode, I'll show you what's behind those numbers. Ready?